In this video, I'm going to walk you through the airways, and clearly I start with the nasal cavity. In this model, I'm looking at the right nasal cavity. The opening to the nasal cavity is called nostril. The space inside would be the nasal vestibule, and I see that each nasal cavity is connected posteriorly to pharynx, and the opening of nasal cavity into the pharynx is called carina. The structures that form nasal cavities are some of the hyaline cartilages that form the anterior portion of the nose. Then we have bones such as nasal bones, frontal bone, ethmoid, sphenoid. And then when I focus on the floor of the nasal cavities, I notice the palate. The front of palate is hard palate. We call it hard palate because it's formed by bones such as maxillary bones and palatine bones. The posterior portion of palate is soft palate, which is an arch-shaped muscular partition. It does not have any bone. Then I see on the lateral wall of the nasal cavity, I do have three elevations. These are nasal conchae, that's superior nasal concha, middle nasal concha, inferior nasal concha on the right side. Recall that superior and middle nasal conchae are part of the ethmoid bone. However, inferior nasal conchae are facial bones. When we analyze the mucous membrane that lines the nasal cavities, in most of the mucous membrane, we do have goblet cells in the epithelial tissue, except in olfactory epithelium. Olfactory epithelium is a very small part of the epithelium right here, very close to the cribriform plate of ethmoid bone. That's the only part of the epithelial tissue that lines nasal cavities without goblet cell. However, we know olfactory epithelium contains those receptors that detect odorant molecules, and we know that cribriform plate of ethmoid bone has those olfactory foramina that are pathway for olfactory nerves to move from nasal cavities into cranial cavity. Below each one of these conchae, we do have a groove. We call these grooves nasal meatuses. So we have superior, middle, and inferior nasal meatuses. Having these projections and grooves increases the surface area, so that helps us in many different ways. First of all, increasing the surface area means we have more epithelial tissue. For example, we have more goblet cell, more secretion of mucus, and also aside from mucus being able to trap debris and particles, we know that mucus contains water. When air enters the nasal cavities, we have to add a little bit water to air to make it moist, to reduce friction. Also, we receive extra mucus from the paranasal sinuses, which in this model we see only two of them. We see frontal sinus and sphenoidal sinus. However, recall that also in bones such as ethmoid and maxillary bones, we do have paranasal sinuses. These are very small spaces filled with air that we have in frontal, ethmoid, sphenoid, and maxillary bones. And they all have ducts opening into the nasal cavities and secrete extra mucus into the nasal cavities. And aside from receiving extra mucus from the paranasal sinuses, we know that exactly below the inferior nasal conchae, we have the opening of nasolacrimal duct. That's the pathway that brings lacrimal fluid here and empty it exactly here, and then lacrimal fluid get mixed with mucus. In general, not losing the water is also something that having these conchae and meatuses could help us with, because every time that we exhale, these elevations and grooves help us to trap water droplets and not lose too much water, to not get dehydrated. Now I move to pharynx, which is a funnel-shaped tube. It's about 13 centimeters, and we divide it into three parts. The upper part, which is behind nasal cavities, up to soft palate, is called nasopharynx. Then the part that is in the middle and is behind oral cavity, from soft palate to the upper edge of epiglottis, that would be oropharynx. And the most inferior part of pharynx is laryngopharynx. Note when I get to laryngopharynx, I have two choices. Either I can go posteriorly and enter esophagus, which is the pathway for food and drink to get eventually to stomach. Or from laryngopharynx, I can go anteriorly and enter larynx. Larynx is the pathway for only air. When I analyze the three sections of pharynx, I notice that nasopharynx is pathway for only air. However, food, drink, and air pass through oropharynx and laryngopharynx. So it is very important that when we swallow food and drink, we make sure that food and drink don't get into larynx. Exactly at the moment that the mass of food and drink get into laryngopharynx, both pharynx and larynx move up, and epiglottis, which is one of the cartilages of larynx, and it happens to be elastic cartilage, quickly moves down and closes larynx. It stays closed until that mass of food and drink gets into esophagus, and then it uses its elasticity to jump back up and opens the airway again. So that would be the job of epiglottis, to prevent food and drink to get into our airways. I go back to nasopharynx to analyze a few anatomical structures that I have here. I notice the opening to auditory tube. That would be the tube that connects tympanic cavity in middle ear to nasopharynx. And through this tube, we exchange a little bit air to equalize the pressure of air between tympanic cavity of middle ear and atmosphere. Then I see on the back wall of nasopharynx, I have a very large tonsil, which is called pharyngeal tonsil or adenoid. Obviously, that's part of the immune system. It contains lots of immune cells, including some immunocompetent cells. When I move down to oropharynx, here I can find two pairs of tonsils. I can find palatine and lingual tonsils here. So that would be the right palatine tonsil, and this is the right lingual tonsil, which happens to be inside the base of the tongue. Clearly, the location of these tonsils tries to guard these openings. Then I move down to laryngopharynx, and because we're focused on airways, obviously I have to enter larynx, which is a very short tube. Basically, larynx extend in front of C4, C5, and C6. It's very short, and it has 9 pieces of cartilages that in a separate video I talk about each one of them individually. In this model, I can see only epiglottis, and I see epiglottis through a ligament is attached to the largest cartilage in larynx, which is thyroid cartilage or Adam's apple. Clearly, thyroid cartilage of male is much larger and more prominent than a female due to being stimulated by, for example, the male sex hormone testosterone. And the last cartilage that I have in larynx is cricoid. When I get here, larynx ends. However, before I move down, I would like you to pay attention to these two folds that we see on the lateral wall of larynx. The upper one is vestibular fold. We use the 2 vestibular folds that we have to hold our breath voluntarily. For example, let's say if there is a bad smell in the room. But the lower folds are true vocal cords. These are the ones that we use to actually generate sound. And when sound is generated here, then sound travels through all the pathways that I discussed so far. And those pathways all help amplifying the sound. That explains why whenever we have inflammation in any of these air pathways, our voice changes. Then I noticed that larynx connects laryngopharynx to trachea. And when I follow trachea, I note that trachea has 16 to 20 hyaline cartilages. Recall, hyaline cartilage is strong, and at the same time, it allows a little bit flexibility. Having so many hyaline cartilages in our air pathway allows airways to stay open all the time. Without these hyaline cartilages, for example, trachea could easily collapse. And then I move down to the end of trachea, exactly before trachea bifurcate and forms the 2 primary bronchi. I go deep inside trachea and I analyze the mucous membrane that lines trachea exactly in this spot at the end. And I notice there is a ridge in the mucous membrane at the end of trachea, which we call that carina. And when we analyze the mucous membrane of carina, we see that that's the most sensitive mucous membrane in the respiratory system to particles and debris. It simply means throughout all the air pathways that I discussed so far, obviously we filter, we clean air, but if particles and debris get here, the sensitivity of carina quickly initiates a coughing reflex. Basically, the cells that we have in carina can communicate very rapidly with the coughing center that we have in medulla oblongata. Clearly, this is an attempt to bring those particles out instead of letting them to move into the lungs, simply because each primary bronchus send air in and out of lungs. I have two primary bronchi, but I note that the right one is wider and more vertical. The left one is narrower and has a curve. That explains why when we have an aspirated object, it mostly, in most cases, moves to the right side. And primary bronchus enters the lung exactly at this depression that is called hilum of the lung. And then inside the lungs, we see that each primary bronchus divides into secondary bronchi, or we call them lobar bronchi because they supply the lobes of the lungs. And it's important to note that right lung has 3 lobes, so it must have 3 secondary or lobar bronchi, but left lung, as you see, has only 1 and 2 lobes. So left lung has only 2 secondary or lobar bronchi. And then I see secondary bronchi, each one of them divides into several tertiary or segmental bronchi. We call them segmental because the tertiary bronchi supply the segments of the lung. When we say supply here, we mean they let air go in and out of the segments. And segmental bronchi are the last bronchi that we have in bronchial tree. We call it tree because it really looks like tree, simply because the tertiary segmental bronchi divide into narrower pathways that we call them bronchioles. And when I follow the bronchioles, I see in this model the last bronchioles that we see with naked eye without the use of microscope are terminal bronchioles. When we say terminal, terminal means end, that simply indicate that terminal bronchioles are the end of the conducting air pathway. When we say conducting, simply it means that these are pathways only for air to pass through, and while air passes through them, obviously air will be cleaned a little bit. They add to the temperature of the air, they make air moist to reduce friction by adding water. However, in none of the pathways that I discussed so far, gas exchange between these pathways and blood is going to happen. So those structures that we exchange gases, oxygen, carbon dioxide with blood, are located after terminal bronchioles, which I'm going to talk about them later. But for now, I would like to analyze the structure of the lungs. I can see that each lung has a pointy part, which that would be called apex. And it's interesting that the apex of the lung sits a little bit above clavicle. That's basically the only part of the lung that could be palpated. Exactly opposite apex, I have base of the lung that sits on diaphragm. And I note that the diaphragm on the right side slightly is elevated to make room for liver that sits exactly in this spot. And one other thing that I notice is that the right lung is wider and shorter. Left lung is a little bit longer but narrower. And the next I notice this depression in the left lung. That's exactly where the apex of the heart sits. And we call this depression cardiac notch. So it's very easy to tell which lung is left, which one is right. The one that has cardiac notch is the left one. The surface of the lung that faces the ribs and costal cartilages, we call it the costal surface. The surface of the lung that faces the mediastinum region, that would be referred to as mediastinal surface. So as you can see, we find, for example, hilum in the mediastinal surface of the lung. And then I note a section of the ribs. I see the intercostal spaces between the ribs that are filled with intercostal muscles. And next thing that I note is that basically each lung sits inside a pleural cavity. Pleural cavity would be this very narrow space which is between visceral pleura, which is exactly attached to the lung, and parietal pleura. Parietal pleura is the serous membrane that lines the pleural cavity. So pleural cavity is exactly the space between visceral and parietal pleura, and is filled with serous fluid, which is a watery fluid. Having a little bit watery fluid between visceral and parietal pleura obviously reduces friction and allows these serous membranes to slide easily, move easily without too much friction. Now I use microscope and I want to see exactly what I have after terminal bronchioles, and I note that when I move down, the amount of hyaline cartilage decreases. So I move to the microscopic structures. Basically, I use microscope to see these structures, and I see that in the last bronchioles that we have, which we call them respiratory bronchioles, I no longer have any cartilage. They're so narrow and small that literally there is no room for cartilage. Instead, they have more smooth muscles. That would be the responsibility of these smooth muscles to keep these airways open. And then I can see lots of tiny microscopic balloons that we call them pulmonary alveoli. And I would like you to pay attention to these capillaries that wrap around the pulmonary alveoli. We name them pulmonary capillaries. And recall that we know that the blood vessel that branches off and form capillaries would be arteriole. So here I can see a blue blood vessel and a red blood vessel. And I recall that when blood gets here, it is deoxygenated, and basically the alveoli, pulmonary alveoli, and these pulmonary capillaries exchange gases. It means from the inside of pulmonary alveoli, oxygen diffuses into these capillaries, and from these capillaries, carbon dioxide diffuses inside the alveoli. Blood becomes oxygenated and then it leaves. So that makes it clear that this blue blood vessel that I see is a pulmonary arteriole. I get here from arteriole, I get the pulmonary capillaries. I see that gas exchange basically happened. Blood is now oxygenated. And from these pulmonary capillaries, blood goes into pulmonary venules that take blood out. Eventually, the pulmonary venules join together to form the four pulmonary veins that take oxygenated blood back into the left atrium of the heart. I hope that you find this explanation helpful.